I'd like to thank the organizers for giving me the opportunity today to tell you a little bit about some of the work that we do in our group. So we study obesity, and as you might know, obesity has become a global health epidemic. In the United States, more than 66% of individuals are overweight and 33% are obese. Obesity affects more than 1 billion people worldwide and accounts for 17% of the healthcare costs in this country. And unfortunately, we have a very poor understanding of its etiology and few effective medical treatments. Concomitant with this increase in obesity, we're seeing an increase in obesity-related metabolic complications such as diabetes, dyslipidemia, hypertension, coronary artery disease, and fatty liver disease. But very interestingly, not all obese people develop these complications. And when they develop these complications, they develop different ones. Unfortunately, medically, we treat these people the same, diet and exercise, which is highly ineffective and leads to high personal and medical costs. And so it's critical to define the causes of obesity and its complications to improve diagnosis, care, and reduce costs. And as people gain weight, they can deposit fat in their liver to cause non-alcoholic fatty liver disease. And this can be in the form of steatosis, which can progress to inflammation, scarring, and cancer. And just like obesity, this has increased over time to about 24% globally. And then to measure this, you can use histology, but really this isn't used clinically because it's so invasive. And so many times it gets picked up because people have elevations in their liver function tests, but these are not sensitive or specific but generally they need to be followed up with imaging. And because of this, more and more of this has now been diagnosed. And NAFLD has now become the #1 cause of liver disease worldwide. And like obesity, we have no effective medical treatments, making it now one of the most unmet medical needs of our time. And just like obesity, only some people deposit fat in their liver. So even bariatric patients, only 90% of them deposit fat in their liver, 10% don't. And only a fraction of them go on to get liver disease. And in work we did, individuals that have 30% fat in their liver, 66% of them meet criteria for having the metabolic syndrome, which is high triglycerides, low HDLs, high abdominal obesity, insulin resistance, and hypertension. And some of them go on to get cardiovascular disease. But currently, medically, we have no idea who's going to deposit fat and then get liver disease versus cardiometabolic disease. And this is what we're interested in finding more about. And to study this, we've taken a human genetic approach, and both obesity and fatty liver are common genetic traits, which means they're genetic variants that when combined with the environment predispose disease. And then we and others have helped establish that overall obesity by BMI, abdominal obesity by waist to hip ratio, and NAFLD by steatosis are heritable and can be studied in the population. And the reason why we took a human genetic approach that I want to emphasize is that if you can identify genetic variants that associate with a disease, by definition, they're causal. And that's different than many other associations like money and power, where you don't know which one caused one. And the reason for that is your genome came before any phenotype that you could have developed. And this is very powerful because it allows you to use what you find to understand the susceptibility of a person to disease, to define the causal influence of that disease, define personal risk, and develop tailored therapeutics for that disease. And when we started this was just a pipe dream. And in fact, the first 3 years of my postdoc, nothing replicated in any of the studies that we did. But eventually, we were able to get reproducible associations. And really, one of the key factors to this was power and increasing sample size. And over time, we've been able to identify genetic variants. These are SNPs and associations with BMI shown here. And you can see these very reproducible spikes coming off of this, which is reproducible associations. But to do this, we really had to do a lot of development in terms of advancing genotyping platforms, phenotype annotations, and harmonization, genotype annotations and harmonizations, QCing of data, which I won't talk about. But really, this has been highly successful over the years. And now in our published and unpublished findings, we have over 9,000 variants that associate with measures of obesity and over 400 that associate with liver function tests or fatty liver disease. And but one of the challenges, as you heard previously, is that when we find these associations, sometimes they land in between genes. And the other big challenge is actually most of the genes in the genome, we don't really know how they work. And so to get around one of these, we helped develop one of these bioinformatic methods. And the idea behind this is we may not know what a gene does, but if it transcriptionally goes up and down with other genes that we know what they do, now you can sort of put it in a biological context. And so what Toon and Muda led is that they downloaded cell expression and tissue expression data sets, and then they did principal components across those, and then mapped gene sets to those, and then mapped genes to those. And then what you can do is that you can drop anchor from your GWAS and look at the genes underneath it. And then you can look to see if there's enrichment in particular tissues for those, if particular gene sets are enriched in your associations, and particular genes at different loci link up by being in the same gene set. And this really dramatically increased our power to sort of see sort of connections between genes in our GWASs. And when we applied this to our genome-wide association analysis of BMI, the nervous system really came out as very significant. And again, we were expecting to hit the Krebs cycle, but really we hit the nervous system. And this was actually very, very important because BMI has a stigma associated with it, which is that it's all willpower. But really the fact that we're able to map this says that it's biological. And in fact, we can map it to the nervous system, which is really fantastic. And then when we did it for abdominal obesity, we got more fat biology. And when we did it for liver function tests most recently, we can see the liver as well as the intestine, pancreas, and adrenal, because these are, as I told you, not completely specific for the liver. And then when we looked at pathways that come out for BMI, we could find highly enriched pathways that affect polyphagia, which is overeating, energy metabolism, and neuronal biology. When we did it for abdominal obesity, we could find mesoderm development, cell growth and survival pathways, and glucocorticoid receptor signaling. And when we did it for liver function tests, we can find lipoprotein metabolism, triglyceride metabolism, and arachidonic metabolism. And this likely represents a combination of non-alcoholic and alcoholic fatty liver in the population. So this is really a pathway analysis that was not previously possible, and the bioinformatics methods really allowed us to now hone in on some of these pathways as being important. And then they also allowed us to identify specific genes that could affect these processes. So one of the genes in our common obesity data set that we landed near was something called MC4R. And this was actually previously known to be a monogenic obesity hit. And then since that time, they've developed MC4 agonists for the treatment of this rare genetic obesity. And now this company is going to the population level and finding individuals that have common variants that associate with obesity and seeing if that drug would be effective for them as well. In addition to that, we hit a lot of genes, and in fact, the majority of our genes, we have no idea how they connect to obesity and fatty liver. And one of those was like the calcitonin receptor at the time. And really, in work done by Toon in combination with Martin Myers here, they mapped different neurons in the dorsal vagal complex that can affect appetite. And there's a particular kind of neuron that has the calcitonin receptor. And when you stimulate this, it actually causes appetite suppression. So this is a new type of neuron and a new target for obesity that could potentially be better than some of the current targets, which is GIPR and GLP-1 agonists that currently do suppress appetite, but actually have a lot of side effects like nausea. And hopefully these could be newer, better targets for obesity. And finally, our first and best hit for fatty liver disease is PNPLA3, again, a novel gene that was now connected to this disease. And really nice work done by Helen Hobbs. She's shown that this is a gain of function that prevents sort of the release of triglycerides from these lipid droplets, which somehow accumulate and cause damage in the liver. And again, this is now being targeted with ASOs for the treatment of non-alcoholic fatty liver disease. So really, this work has led to actual treatments that are now in place for obesity and non-alcoholic fatty liver disease that are being tried. But then, as I told you, the majority of the things that came out of these screens are totally novel. So in our lab, what we've done is we set up cell and mouse models, and then we've knocked down and overexpressed these genes and looked to see whether they accumulate fat, as well as look at other biochemical properties of altering these genes. And we've also progressed to genome-wide CRISPR screens to look for genes that may have not been picked up by GWAS, but certainly can affect these phenotypes, and then are now adapting these to high throughput, high content screening to be able to do this, to be able to find new pharmacologic and targeted treatments for these conditions going forward. In addition to that, so as I told you, we carried out some of the largest genome-wide association studies for liver disease and liver function tests. And then we looked to see, how many of these are actually fatty liver specific by crossing them to an old data set that we had on imaging. And we did find that a fraction of them, but only a fraction of them actually associated with non-alcoholic fatty liver disease. And so there was still a great need to find more, variants that associate with non-alcoholic fatty liver disease specifically. And so to do this, we increased our sample size to be able to do this. And so we assembled over 23 43,000 samples that had computed tomography measured fatty liver. And then from the UK Biobank, we had 43,000 MRIs available, but we didn't have fat measures on these. We only had 4,600 of with fat measures. So what we did is we downloaded the raw MRIs. We segmented the liver, and then we trained a deep neural network, ResNet, to be able to measure the PDFF, which is the fat in the liver. And then we trained it in a certain portion of the samples and then tested it in the rest. And we got it to work very well with a correlation of 0.97 for reading these. And then we applied it to read all 43,000 of these. We then carried out genome-wide association studies across these imaging data sets and combined with doctor-diagnosed fatty liver disease and 3 other data sets with a fixed effects meta-analysis, identified 17 independent variants. And we used some of our algorithms to refine some of the hits here to ones that are specific for fatty liver. And then we took these hits and then created polygenic risk scores, which is to take the gene weighted by its effect size and look to see if we could identify individuals that are at high risk of having fatty liver disease, cirrhosis, or hepatocellular carcinoma in an independent data set, which is the Michigan Genomics initiative from here. And what you can see is that these polygenic risk scores are able to identify 10% of individuals with an odds ratio of 2 and above, 5% of individuals with an odds ratio of greater than 3, and 1% of individuals with greater than 4 odds ratio of 4 for having these diseases. And really, this is the best non-invasive biomarker of having a risk for these diseases that's currently available, which is very exciting and much better than anything that we've had before. And in work that I don't have time to show you, we've also shown that these can predict incident disease, which is new cirrhosis, and then these provide information above and beyond these clinical markers. So we're hoping that these can be implemented to better risk stratify individuals for better care in the future. In addition to that, we looked at how these 17 SNPs associated with multiple diagnoses and traits across the UK Biobank, and this is increase or decrease of these. And basically by K-means clustering, that they fell into 7 different groups. And then again, we made polygenic risk scores from these and then looked to see how they affected multiple human traits. And what you can see here is that all of them affect steatosis, because that's our study was biased for that. But then some of them increase cirrhosis risk and some of them don't affect it. Some of them increase serum triglycerides and lipid traits and increase your risk of heart attack, and some decrease it and some increase it. Some increase diabetes and body mass index, whereas other ones decrease it. And really what you see here is that 7 different patterns of these 7 metabolic subtypes, and these actually don't all match the epidemiology right now of fatty liver disease, which has this pattern here. And the reason why this is important is this comes back to the idea is that epidemiology lumps everybody together, but it's highly inaccurate. And as I told you, not all obese people develop complications, and then not all people deposit fat in their liver and get complications. And so what this now allows us to do is now we have biomarkers for these phenotypes, these metabolic diseases. And not only do we have biomarkers to identify these subtypes, but we can match them back to targets for therapeutic intervention. And you can see that depending on the target, so if you have, for example, if you want to target PNPLA3 to decrease fat, you're going to increase sort of your heart risk. And if you happen to unfortunately apply this drug to these people, you're actually going to increase their side effects from this medication because they're already predisposed to heart disease. So really, this can help you to find the individual and match them to the right drug. In addition to that, when you look at these, they fall into nice new pathways. And some of these, like PNPLA3 with its other members, cause a fatty liver by decreasing lipoprotein output. Some cause it by increasing lipoprotein input to the liver to cause fatty liver. Some increase risk of diabetes and insulin resistance peripherally, and maybe by increasing fatty acid release to the liver, increase fatty liver. Some may affect insulin signaling in the liver to promote fatty acid synthesis, including by taking glucose and making it into fatty acids. And some may function by actually not allowing triglyceride to exit the liver and diverting it to other kinds of phospholipids that can cause fatty liver disease. So this gives us new insights into the pathophysiology of the disease. And in fact, we can now better link these to ongoing treatment trials like the ASOs for PNPLA3, to certain thyroid agonists that affect lipoprotein buildup, to ASOs that decrease triglycerides, to diabetes-specific and MTTP-specific treatments, and then specific treatments for de novo lipogenesis, depending on the pathogenic mechanism that the patient's really suffering from. And finally, we are taking this to the clinic, and here what you see is that a few years ago we found that insulin resistance multiplicatively interacts with PNPLA3 to promote fatty liver disease. So if you have the high risk genotype and high insulin, you have a greater than a 90% chance of getting fatty liver. Low risk genotype and low insulin resistance, 20%. And the people that are at highest risk genetically actually benefit more from decreasing their insulin resistance. So we're actually recruiting from the Michigan Genomics Initiative and putting them on keto diets, and we're measuring how much fat accumulates in these individuals with the assumption that these guys are going to benefit more than the people that are at low risk. And again, you can see how we can take these genetic information and then use it to personalize treatment for future. So basically, I hope I've shown you how we can use human genetics to define disease causes and guide precision treatment. We can use population science to define causal genetic variants and environmental precipitants. We can use integrative science to define causal genes, pathways, tissues, disease subtypes, and tissue targets and side effects. Functional science to test these targets and define mechanism and develop assays for development of treatments, and clinical science for risk stratification and genetically based diseases. And really, I think we've come a very long way in the 10 years, as I just shown you, and I see a very bright future with therapies that in the future, based on this work, will be more effective with fewer side effects. So I'd like to thank our participants, many of the consortia that gave data for these work. I'd like to thank the GOLD Consortium for the fatty liver work, especially Nicole Allred, who co-leads this with me, members of our laboratory and our funding. And I'd really like to thank Gil, Brian, and my colleagues in DCMB that have provided a wonderful home to do this work over the last 10 years. Happy 10th anniversary, and many more to come. Thank you.
Yeah. So our original plan was to have a 15-minute sort of Q&A to invite the speakers back up and just have a panel discussion. It turns out we're about 15 minutes behind the schedule. So I think instead we're going to go straight to break. I would encourage you, if you have questions about individual talks, to approach the speakers and ask them during the break. We'll reconvene here in about 15 minutes and maybe thank the speakers one more time.
Oh, hi, Kurt.
Nice to meet you.
Nice.
I'm so sorry that we didn't show up earlier.
Okay.
I think there was a miscommunication, and I thought that you wanted me here at 2:30. So I think it was a miscommunication.
It was. Yeah. It is what it is.
We will make sure that
Okay.
we get
So.
When you connected to the Zoom, how did you do that?
I did it through here. We did it through the video conference,
Sure.
right? And we called in. So we did a video conference. We dialed by address. We
Yeah.
went, and then I think I went 2 stores. Then I
Did
went
you have
video
the Zoom
conference.
up on here too?
Pardon me.
So can we connect to the Zoom here? Is this your account?
I'm not on Zoom here. That's not how I was showed to do it by the previous person when I had him.
Wonderful. Oh, like us. Okay.
Oh, I have the.
Hey, how are you? Good, long time. Congrats on the 10-year anniversary. Yeah. Yeah, so for the
Yeah. Maureen and Alla, are you coming? Yeah, come here now because I don't want to be the one in charge of being late. I'll wait for Maureen. So this is a special moment of pride for us. We usually give the student awards during the retreat. These are departmental awards, and we have 3 different kind of awards. So I think Maureen is starting. Yeah, that's how we had it.
So I'm going to be presenting the Bioinformatics Student Service Award this year. So this year, I think it was a no-brainer identifying who would get the service award. We're giving it to 2 students. So back when we became a department, there were not any student-led groups. It's been wonderful to see over the years several student groups formed. So this award this year is going to the 2 students who started our Bioinformatics Graduate Student Association, or BGSA. And not only did these students start this during the pandemic, nonetheless, but they were also involved with other activities as well, like DANG and the PIBS recruitment. So this award goes to Ford Hannum and Kathy Smith. If you can come up to get your award. We're supposed to pause for a picture. Thank
you.
Is Ford here?
Stay here.
He-
We want all awardees and all. Is Ford here? No. Okay.
Oh, okay.
The next student award, and I believe the awardees, just for the audience, are not aware who they are, is for the Academic Achievement Award, which we typically give to a student who, after their second year, passed prelims without any conditions and has a 4.0 GPA. And when there are several contenders, which has happened most of the years so far, then those who took the particularly hard courses. This year, the awardee is Yufeng Zhang. I don't know if she's here. And she's continuing a strong, come here, please. She's continuing a very strong, tradition that was started by our next speaker, in fact. She came to us from Biostat department. And turn it around. And so she has taken very heavy biostat classes and passed prelims without any conditions. Now, okay.
And so the third award is newer for our program. We are very pleased and very proud to recognize master students for their academic achievement. And this year we had two people, so we basically could not decide, and they're both equally deserving. So this year the award goes to two people. The first person is Lingrui Tsai. Lingrui, are you here? Right, congratulations. And the second award goes to Jijun Yu. Not here? Okay.
You want a picture of all of the awardees that are here and us? Yeah. Great. Congratulations for your great achievements and great service for the program. And hopefully Ford and Zhejiang will get their awards at some point. Yeah, yeah. Thank you. So Dan Rhodes is first. Yeah. So the next two talks, I have a great pleasure as graduate chair and director of the bioinformatics program. Our first speaker is one of our very, very first class that graduated in 2006. Daniel Rhodes, who came to us actually initially also wanting to study medicine, but he then quickly decided that research is much more exciting. He worked with Arul Chanayan and then founded a company with him and switched between companies. And I think he will talk more about this. So Daniel, take it from here.
Is this mic there? Oh, well, thank you so much. It's wonderful to be here. Congratulations to the department on 10-year anniversary. Congratulations to Brian, to Gil, and all the faculty and the students. You're part of something special here. When Brian asked me to speak, it kind of took me down memory lane. He asked me to talk about my time here in the bioinformatics program and the career journey that I've had since. So it's a real pleasure to do that with you today. So the title of my talk, and it's hard to believe, is reflecting on 20 years in bioinformatics. It was about 20 years ago today that I joined the program, the bioinformatics program. And so I'll give you a little bit of insight. So let's see here. There we go. Okay, so 3 themes for today. The first is gratitude. A huge thank you to the leaders of this program and for the opportunity to be one of the first graduate students. This experience kind of set me on my journey. And it has really been a continuous theme from actually the first projects I did 20 years ago to what I'm doing today. I'm not saying that that's a rule and you all have to continue to work on what you're working on in your graduate studies for 20 years, but it happened to work that way for me. So a big thank you to the program, the leadership of the program. What I'm going to do in my talk is take you through a few stories. As I was kind of looking back, I've got a few chapters in my career, starting with my time in the program. And my hope is to leave you with a few lessons learned, especially for the students who are in the thick of it right now. To think about how you might best spend your time, what things you might focus on, what perspective you might bring to your work. So I hope I can leave you with some valuable lessons learned. So let's start with stories. And so the chapter 1 of my story in bioinformatics is the University of Michigan Bioinformatics program. So as was just alluded to, I came to the program as a MSTP student. Actually, before I was an MSTP student, I was just a medical student. Very proud to be a medical student at the University of Michigan, but I found out quickly that I really wasn't in love with the idea of practicing medicine. I loved the science, I loved the problems, but didn't love the idea of practicing medicine. And I admire those who do. All of my former classmates, lots of them I keep in touch with, but I've kind of found it really just wasn't for me. But at this time, sort of late 90s, early 2000s, we've got the human genome, and now we've got a technology which is now like this old time technology, DNA microarrays, and we're beginning to generate for the first times enormous amounts of data. We didn't call it big data back then. We didn't really have that term. But for me, this prospect of using something that I always felt gifted at, mathematics, and the big data and genomics to unravel complex human disease was just kind of the coolest thing that I could imagine working on. And so kind of the first kind of lesson learned for me is this was like big light bulbs going off, like intersection of what I'm good at, what's really interesting to me, and what matters to society. And so with those things coming together, I really felt like I found my calling and passion. And so I would encourage each of you, when you're thinking about the work you're doing, make sure you find work that lines up with your passion. Don't get trapped in kind of incremental projects or a space that you're just not that interested in. Even if it sets you back, even if it puts you behind, find the area that you're passionate about. So like I said, I transitioned from medical school to the medical scientist training program. This was sort of my kind of half step out of medicine. I'll tell you in a bit how I ended up taking the full step out of medicine, but joined the bioinformatics program and was just so fortunate to meet and then study with Dr. Rul Chanayan, who of course now is one of the greatest cancer genome scientists on the planet and has just had seminal contributions. Way back when I was a Rul's first graduate student, he had just published a Nature paper on prostate cancer genomics. I had read that paper with great interest and sort of knew right then, if I liked this guy, this is who I was going to train with, because he was just doing incredible work. My first meeting with a Rul, having had some experience coding and doing mathematics, and telling a Rul, I don't really think I'm going to be that good in the laboratory, but this data problem is something that's for me. In that first meeting, when I was just considering working with him, he said to me, what if we tried to put together all of the world's published cancer genomic information? What could we do with that? And I left thinking, oh wow, I mean, this is like a dream come true. Like I could not only advance the field and advance our scientific understanding of cancer, but also could build something that had impact. And so it was that first meeting that set me on my journey with a Rul. And I remember thinking, I like this guy because he thinks big. A Rul is not an incrementalist. He's looking for the next big challenge, the next big opportunity to advance research. So I've kept that kind of feeling I had after that first meeting with me for my whole career. Like, think big, let's do things that really matter. So as a graduate student now, having committed to a rules lab and to the bioinformatics program, finishing those first years of medical school, which were somewhat painful for me, but then moving into graduate studies, I found that this was like kind of the most incredible opportunity you could imagine. Your only real job as a graduate student is to dive as deep as you can into a domain, into a space, and become a true expert, and then look for opportunities to make an original contribution yourself. And so I did that, and I loved it because I had found an area that I was passionate about. I consumed everything. When I have time, I still do today. Sometimes I rely on some of the folks on my team to do the consuming and then share with me. But back as a student, I consumed everything. I read every paper that was relevant to our space. Then just read the papers. I read every detail of all of the supplementary information. And I studied it so that I truly understood the work that author had done to deliver their scientific impact. I think too often, students take a light look at others' work and don't go all the way in. And I would encourage you, go deep, study the supplementary information for the leaders in your field, for the great work that they've done and published. Make sure you know it almost as well as they do, because then you can take the next leap. So you'll only ever be able to do this in your career as a graduate student. As you move into your career into faculty positions or into industry, you'll never have the time and space that you do as a graduate student. So take advantage of it and use that time. Okay, so in a rules group, we did incredible work on cancer genomic data, but we also said, let's not just do bioinformatics for ourselves. Let's build something. So we built a cancer genomic database and web application called Oncomine. So that grand challenge that a rule gave me at the beginning, what if we could assemble all the world's published cancer genomic information? We did that, and we didn't just do research, we built a product really to enable our research, but equally importantly, to enable the research of others. So when I think back to that work in building Oncomine, I learned how to build data models, how to build data curation pipelines, how to build data analysis pipelines, statistics, and then also how to build software. I didn't do all that work myself. We had a team, but I learned how to go from a concept and data to product. And so this is another thing. Now, this isn't going to be right for everyone, but I would say when you have the opportunity, don't just do bioinformatics for your own research. To the extent you can, build something and share it with the world, because if you can, you'll just absolutely amplify the impact of your work. And that's what we were able to do with Oncomine. Now, a rule is well-resourced laboratory, right? So I can't say that, I can't take that for granted. So when we were building Oncomine, we had scientists, IT folks, database administrators, web application developers, and data curators, all as part of the team. And so that's another thing I learned as a graduate student is you can't do it all alone. You really need to find others who have skill sets that you don't have and bring together a team around a common cause. And that experience I had building that Oncomine team is something that really carried through to my career in building companies. And so it really, as Bo said, it really is all about the team. So with that Oncomine experience, we grew that application to ultimately have tens of thousands of users globally. I always had an entrepreneurial bent. I always wanted to build a company, even from early days in my PhD. And so we connected early on with the Office of Technology Transfer. So this is another just point to make. If you're doing work that's leading to new software applications or new intellectual property around diagnostics or therapeutics, we have an incredible Office of Technology Transfer here at the University of Michigan, and they will partner with you shoulder to shoulder to understand the novelty of your work and to file patents, to file copyrights and trademarks and the like. So we did that in the early days of Oncomine. We said, hey, we're going to make this resource available to all academic researchers free of charge for not-for-profit research. But we're going to require pharma companies to license access through the University of Michigan. And this brought in funding for the laboratory and also showed us that we really had a business opportunity here. And so I was able to lean on the Office of Technology Transfer, Rule and I were, to advance this ultimately to what became chapter 2 for me, which is spinning this work out and creating a company around it. I would be remiss if I didn't look back and think about using the Oncomine database together with Scott Tomlins and Arul to, pardon?
5 minutes.
Yep.
Got a lot of stories in there.
Well, I think we're just going to go right to chapter 2, 3, 4, Brian. Thank you. This is the fun part anyways. So we discovered gene fusions from the database and then went on and won the AACR Team Science Award from that. So it's something we're all quite proud of. After Michigan, we spun that work off and I transitioned from graduate student to CEO. Very interesting transition. Expert in the data and the application, but really knowing nothing about building a business. My advice to anyone who has that entrepreneurial bent is take advantage of this incredible community we have in Ann Arbor. Tech Transfer, Ann Arbor Spark, and all sorts of programming inside the University of Michigan. Fast Forward Medical Innovation, the MTRAC program, the Michigan Biomedical Fund. You have this incredible community just waiting for brilliant students and faculty to take their technology out and create companies around it. And they're just really waiting to help you do that. So we were able to do that with Compendia and build a really incredible company. A company was acquired. This is a process as well. And so moving from Compendia, small startup, that was my thesis work, to a Fortune 100 company in Thermo Fisher Scientific, incredible opportunity to build diagnostics and lead teams. And this is really where I learned how to truly lead teams and how to be a leader in an organization. Incredible mentors, and just really emphasized for me the importance of leadership. We did a lot of great work there. Ultimately, that kind of moved my work out of the kind of theoretical data domain and into the physical world of diagnostics and delivering real value to patients, which has really been central to my personal mission and what I'm working on and delivering. So that leads me to where I am now. So this is my second company called Strata Oncology, where I'm the CEO. We're a precision oncology company. We're sequencing tumor genomes and transcriptomes to drive personalized treatment. And we're really trying to take the next leap beyond tumor mutations and into multivariate DNA and RNA predictive biomarkers for cancer therapeutics. Been an incredible experience with incredible co-founders and investors, and now delivering on what we think is a really big vision. So this is kind of last lesson learned is this idea of visioning. It works. Create a vision for yourself, for the work that you're doing, for your team, and really try to detail that clearly. And you can really make kind of anything you dream up come true is my real belief. All right, so gratitude, stories, lessons learned. I've got the list there. This is what I took from being a graduate student in my career path. I hope it was helpful to the students here. Special thanks to those listed, and I'll stop there. Thank you.
Wow, thank you for this inspiration for students. I don't think you were there in the lunch run with students. There were several students talking about entrepreneurship. So those who asked, please go to see Daniel, who seems to have had a lot of success and a lot of experience. The second student alum from the program to introduce is Aaron Shellman, who was there during the lunch and had already, I kept forgetting to count how many different companies, but she's going to talk about that. Erin graduated in 2012, so exactly 10 years ago. And as far as I could tell, she is the only student who graduated from Chuck Barant's lab. So if anyone in bioinformatics is interested in, Chuck Barant has lots of students, but rare bioinformatics, the first and only bioinformatics student. She also started a trend that you heard about earlier. She came to us from biostatistics, and that trend has continued. Every year we have 1 or 2 students from biostatistics choosing bioinformatics because we are more applied and they're more interested in the applications than in the math. So I don't want to take more of your time. Aaron, it's a pleasure to have you here. Thank you.
Hi, thanks for that introduction. And I really want to thank Brian for the invitation to come back to Ann Arbor and participate in the celebration of the 10-year anniversary of the department. It's quite an honor, and it's been a real pleasure of just walking around and seeing our old neighborhoods. This is a little bit unusual for me. I usually give technical talks, but today I want to step back a little bit and take some stock and share a little bit more about my journey, and in particular, in my background and how I got here. But I also want to highlight how this department really nurtured my interests coming in and developed the skills that have been really critical for my career. Let's see. So I don't want to get yelled at by Brian. I don't know if you see him up here, like police in the time. So I think I'm just going to skip this. This was going to be my in a nutshell, but I'm going to dig into the details anyhow. So for the sake of time, I'm going to move on from the overview and actually take you back, if you can imagine, a little more than 10 years ago, the version of me at Case Western Reserve University in Cleveland. Before I came to the University of Michigan, I was a young economics major. And I was always really interested in quantitative ways to describe human behavior. And in particular, I was interested in this field of behavioral economics, which combines psychology with economics and really seeks to explain why it is that weird humans tend to act really differently in reality than what we would expect using classical models. And if you know anything also about Case Western, it might not come as a huge surprise that at the time I was also really interested in computers. And I included this relic here for those of you too young to remember what life was like before you could carry computers around in your pocket and in your backpack. They used to be pretty stationary, and this is actually a pretty sleek one in comparison to how they sometimes looked. So this is pretty early internet days, but I was already kind of developing this interest in using the internet and computer technology to ask research questions and to collect and analyze data. And so I spent a lot of time in my senior year doing my honors thesis in scraping data from eBay and doing online surveys, building those because it was very actually kind of difficult to do at the time, to explore differences in risk perception and risk aversion across genders. And so very interested in this area. And actually economics was my launching off point in my introduction to biology, and specifically through the study of game theory, where I was introduced to this book, Evolution in the Theory of Games. And this was my gateway into ecology, into evolutionary biology. And as soon as I realized that I could use and translate what I had been learning, the models and the quantitative approaches that I had been learning in economics, and then apply those to this, for me, kind of new field in biology, I was really hooked on that. And I quickly added this evolutionary biology degree to my studies. And then I had the really good fortune between the summer between my junior and senior years of getting an internship at the National Institutes of Health in the Division of Computational Biosciences. And that was my first exposure to machine learning, which looks really different than it does now. But that's what really cemented for me that this was the area that I wanted to be, at this intersection of quantitative sciences and statistics, machine learning, computer sciences, and then most importantly, the biological sciences. And so I continued and extended my love of research here at Michigan. First in the biostatistics department, like Margaret said, and then in the bioinformatics program. And as a member of the Brandt Lab, I was exposed to a lot of different data sets and research projects that were being led by investigators who were using his metabolomics core lab. And doing analysis on that wide variety of topics and research projects gave me just this incredible quantitative foundation, which was really the basis of my career, especially early in my career when I was doing a lot of technical work still. I got a lot of really good practice in developing efficient analysis workflows and making compelling visualizations that tell stories with data, in presenting results, and overall just learning how to be an effective collaborator on other people's research. And those things are still skills and techniques that I use every day in my work. And then in my thesis work, kind of like what I was doing in my thesis work at Case, I again took inspiration from the high-scale algorithms and approaches that were powering the web at the time. Things like Google's PageRank. But instead of mining hyperlinks or social networks, I was applying these algorithms and these approaches to metabolic networks and mining those for structural properties that I could correlate with physiological function. I even used those network properties to explore evolutionary relationships and proposed a metabolism-centric way of measuring organism similarity. Somehow remembered that. So after graduating then with my PhD in bioinformatics, I moved out to Seattle, Washington, and I did what all young, aspiring bioinformaticians do, which is immediately get a data science job in fashion retail. Thanks for that. So I started my career as a data scientist in the Nordstrom Data Lab, and we called it a lab, so it's legit. This was a period of really enormous growth for me, and it was actually a really ideal place for me to start my career. Data Lab was kind of a special team. It was this interdisciplinary group that was split between these super experienced, really seasoned software engineers, and then these somewhat less seasoned, less experienced data scientists like myself. And this is really the team where I cut my teeth and I learned how to write and ship code that millions of customers on Nordstrom.com could actually rely on working. And this is where I grew my ability to work collaboratively on a team and appreciate and grow my depth and tools to help me work on joint code bases and have higher standards of practice. And my specialization was recommendation algorithms and personalization. So thematically, again, I found myself working with behavioral data from the web, behavioral and preference data. And so at the time that I was learning a lot about engineering, I was actually showing off a lot of the scientific skill set that I developed here in this bioinformatics department, including obviously the visualization skills of a highly seasoned phylogeneticist, I think. One of the projects that I'm the most proud of is actually what some of these images I was able to dig up are from. And it involved creating an outfit builder that required a lot of creativity and quantitative reasoning and actually empiricism. So our goal here was to create a really compelling recommender to help people build and style outfits based on an initial seed product. So maybe a shirt or a pair of pants that they've already selected. So color is obviously a really critical piece of styling. And we had a ton of really granular data about garment colors from these photos from the catalog. But the question was, how do we teach algorithms to reason about colors and to make stylistic and realistic choices? Well, one approach you might imagine is just use color theory. So we know quite a lot about primary colors, secondary colors, but it turned out that that purely kind of theoretical approach didn't produce really realistic styles like you and I would pick out, or some of us would pick out for ourselves. So instead of a color theory approach, I took an empirical approach to this problem. I realized, look, we have all of these actually amazing domain experts called fashion designers who make these really good decisions about how to combine colors all the time. So why don't we just learn from them and then feed that to the algorithms? So when something like this, we mined every product in our catalog to derive what we called complementary color sets by extracting all of the colors that appear together in the same garment. So you can kind of see that example from that dress up there. You've got 4 kind of colors, those 2 shades of blue and a black and a gray, and together those work together to create the pattern that you see. Then we looked across all of those sets and we quantified the frequency at which those colors co-occurred within garments. And then to make recommendations, all we had to do was apply some simple logic like match this with a contrasting color from the set of commonly co-occurring colors. And we could find contrasting colors. You know, how do you explain what a contrasting color is then to an algorithm? You can do that by using things like distances in the RGB color space, very similarly to how you might navigate evolutionary distances in bioinformatics. So this is a great example of me taking a lot of the approaches that I was using and applying them to a super different setting than where I had learned them. This approach, sort of empirical approach, led to a lot more realistic styles and also meant that our algorithms evolved over time as the data continued to update the models they reflected seasonal styles and trends, which was a little bit of a benefit that we really hadn't anticipated initially. And so we were teaching them to think the way that our designers think. Okay. Okay. See. Okay, let's talk about where I am now, though. So this is just a short transition slide, really to help make the conceptual link. So coming from a really different area back into where I am now as a leader of the data science and data engineering organization at Ginkgo Bioworks, a synthetic biology company. You might think, wow, that seems really different. Well, the reality is, it's not all that different. And actually, a big piece of what we do in this space is also create recommendations. So conceptually, what all you're doing there is taking a big search space and trying to compose it down to relevant results. And that's actually one of the first things that I did at Zymergen, very related to now what I do at Ginkgo Bioworks, which is where I am now. So today I lead the data organization there at Ginkgo, which is the leading synthetic biology platform. Synthetic biology, in my view, is the ultimate industry for people like us who have sort of an engineering mindset and who love working across disciplines. And it combines the latest technologies in genome editing, robotic automation, high throughput screening, machine learning, and data science. It brings them together in what we call biofoundries that engineer cells to make materials and molecules for a range of applications. So at Ginkgo, we work with customers in agriculture, flavors and fragrances, food science, waste remediation, and even in the production of cannabinoids. So this practice of synthetic biology is really organized around the design, build, and test, or Dbtl cycle. I'm curious, is anybody, you guys familiar with this concept, the Dbtl cycle? Okay, Brian is. Well, this is something if you are in this space, you'll definitely learn like the back of your hand. The Dbtl cycle is really for rapid experimentation and iteration to an engineered cell that performs to a predefined specification. So usually some combination of yield and productivity of a molecule of interest. So in the design phase, we're using computational methods to identify targets for genetic engineering, and sometimes using those also to optimize sequence. So those might be through mechanistic techniques that maybe you've used, like flux balance analysis, or those can be kind of black box models from machine learning, or they can be combinations of those powerful approaches. In the build phase is where we're actually building the genetic constructs and transforming the cells so that they express the new engineered inserts. And then in the test phase, we screen the candidates at high throughput and we generate large volumes of data that describe the performance of those cells and making the products of interest. Incidentally, as a statistician, this is one of my favorite parts because this is where a lot of the classical techniques really come to the fore. So a big emphasis on design of experiment, on hypothesis testing, on things like normalization to try to mitigate batch and process effects from the measurement data. And then the learn step is all about synthesis and synthesizing the results from those multiple rounds of experimentation and screening to inform the next design step, but then also to bring up the level of abstraction to another layer so that we can learn about biological properties that we can actually move into a different context, move into maybe a different host cell or a different target molecule, and basically teach ourselves to be better engineers of biology. So that's me in a nutshell. Before Brian kills me, he wanted me to talk a little bit about the modern workplace and what's changing for us and what's important about the skills of today to be successful. And it's undeniable, obviously, that the pandemic has changed a ton about the way that we live and work, and we couldn't have anticipated that 10 years ago. But I'm happy to say that this department really prepared me for that rapid evolution. And first and foremost, if you took anything from my career journey, I hope it's the realization that, like this department and the people in it and the programs, modern problems are super interdisciplinary. No matter whether you're in fashion retail or you're in synthetic biology, you're going to be tasked with these really thorny problems to solve. And sometimes those are very high scale problems, internet scale problems. And your ability to successfully find solutions is really going to hinge on how much exposure you've had to different ways of thinking and solving problems, and then your ability to pluck them out of one context and translate them to where you are, to the problem that you're facing. The other big thing for me, is synthesis of information from all kinds of heterogeneous sources. So when I think about the types of things my team does and the data they're using, like thinking about a process like normalization, trying to eliminate batch effects, that might include data from like operations data from liquid handlers. How long was this plate sitting on the platform before it was actually assayed? And then you have the measurement values themselves coming from the assay. And then you might also fold in bioinformatic context that actually describes what those engineering, what was the goal, what was the biological context around what you were engineering that we can actually use to translate what we discover to other settings and become better biological engineers. So it's really about knowing as many disciplines, knowing the thinking of disciplines as you can, and then synthesizing disparate information. How am I doing, Brian? Okay. Good. So the other piece of this is, of course, a lot of us are purely computational scientists. I know that I am, and so maybe the most obvious change in the modern workplace is the move to remote work. I led a distributed team actually prior to the pandemic at Zymergen, and so that prepared me really well, I think, for leading a remote and distributed team again at Ginkgo. And to me, I get asked this a lot: what are the keys to success in this space? And I actually don't think it's that special, or it's not that different from the keys to success without remote work. To me, it really comes down to outcomes and expectations. So outcomes, it's all about being intentional in what you're trying to get out of every interaction, or even with your work. How do you know when it's done? What's the level of performance that's required to solve this application, ship it, and move on to the next thing? Be intentional and know that before you start. Similarly for communications, what do I want to learn from this interaction? What do I want, and for leaders, what do I want my team to come away understanding after this discussion? And knowing that going in seems obvious, but it's a skill that I think we have to get better at. Likewise, expectation setting is really important. We're working across different time zones. It's I think more important than ever to be super communicative and transparent about the settings that make you successful or not successful. How do you want to work? When are you going to be working? When are you not working? And being explicit about those boundaries, I think, are really critical for this crazy era that we're in. And then finally, the most important thing, I think, is having great advocates in your corner. I feel really fortunate to have always had Brian and Chuck in my corner through grad school, and then that has continued for me throughout my career. So again, thank you both for being there for me and for inviting me.
Thank you all. I'm so proud of each and every one of you. And, you know, Dan was at the kickoff for the party. Yeah. You know, and I have a picture of you there. I promise not to go. And Karen was at our kickoff when we launched the Institute for Data Science. And for me, what's so touching really is to just see how your careers have developed, how wonderful people you are, and how you're giving back and coming back to teach us about, you know, what it's like to make the program you feel like. So thanks so much for being here. Thank you, Maria and Maureen and Ala, and congratulations to the students. We'll find our missing students to pick up their award. But everyone should be so proud. Look at what can happen. It will happen. Thank you very much. Thank you. It's time for a very, very special event, and that is the eighth Omen lecture. You know, we did have a little hiatus there during COVID, and then we picked up one, and now we're here for our eighth Omen lecture, keynote event for celebration. We could be more thrilled all of visiting and spending the whole day generously with us. Bill, I'll give it to you.
Thank you, Brian. Thank you all for being here, and those on Zoom. Hope you're enjoying the whole day. This is a very special treat. We have sort of Renaissance man of science, Holden Thorpe. He's following what's already a quite remarkable tradition of these annual, except for 2 years of COVID, annual OMID lectures. And for those of you who may have been at all of them, or some of them, or none, I thought I'd just show you quickly who the predecessors are. So Leroy Hood, Zach Kohani, Hood is Institute for Systems Biology in Seattle, Zach is at Harvard, and David Shaw from Shaw Research, D.E. Shaw, very remarkable chemist. And then George Post, who's in Arizona, Atul Butte, UCSF, Olga Troyanskaya, whose department I visited at Princeton yesterday. And last in June at our retreat, Brad Malin, and now Holden Thorpe. So we're very, very proud of everyone, and we think we've established a great tradition here, and we're sure that Holden appreciates that as much as we do. Well, I don't need to read to you what's already beautifully described here in the bio sketch for Holden, just to say that he's a world-class chemist, grew up in a small town of Fayetteville, North Carolina, learned from his parents, who was inspired by his parents to be greatly interested in music and theater and the arts. Went to a University of North Carolina, Chapel Hill, and then he did a 5-year PhD program at Caltech in 3 years, Harry Gray, and then he did a postdoc at Yale in about a year, apparently, and then he returned to North Carolina for a faculty position, and soon became, I think, chairman of chemistry, but in any case, the dean of arts and sciences. And from that, at a very early age, became the chancellor, which is like the president of the University of North Carolina at Chapel Hill within the North Carolina upper higher education system. So that was quite a rapid and remarkable and much appreciated early career. And after some turmoil, which I think he probably had no part at all except to shake his head with the athletic department, he relocated to a position as provost at Washington University in St. Louis. And after 6 years there, he was recruited to be the new editor-in-chief of our most distinguished set of journals, Science Magazine, and now the family of Science Magazine journals. So that reflects my hat as once upon a time president and chairman of the board of the AAAS. So welcome, Holden. We're very pleased that you were even so invested with our students and the faculty today, and we're all eager to hear your remarks. By the way, I expect, from the title you already heard, it has a lot of policy content. He's also published 2 books, which are mentioned here in the text with his friend Buck Goldstein. You'll find the titles pretty exciting. Welcome.
Thanks, Gil. Thanks, everybody, and congratulations to your department on 10 years. What an exciting day it is for you, and what an honor it is for me to be among these distinguished folks who have given this lecture. This will be first, Gil, thank you for everything you've done for the AAAS. I agree with you that the Science family are the most distinguished journals in the world, but I've seen a lot of instances of the word nature up here today. So I just want to make sure everybody knows where to find me. Because, as Gil said, you should think of us first. We still might reject your paper, but you should still think of us first. No, listen, if you get to be the editor-in-chief of Science, it's just one of the truly extraordinary opportunities for a scientist, and I'm so fortunate to be able to do it. And I appreciate Gil and his intro mentioning the fact that there was turmoil at the University of North Carolina. I hate it when people say, oh, and then he was the chancellor, and then he was the provost, and like the students are going, oh, I guess everything went perfectly in his life. And that's not a very good educational message. Because even though I had this meteoric rise in my career, I got absolutely destroyed at the University of North Carolina when I was a chancellor, by a combination of politics and intercollegiate athletics and inexperience. And so it's just a reminder that when you hear these intros of people, a lot of times the hard parts get left out, and no one who gets a glowing introduction really has that story. So that's an important thing for students to hear. And I really enjoyed interacting with you all earlier today. So this will be the least technical talk you've heard because I don't do experiments anymore. I don't have a lab anymore. What I do is observe the behavior of scientists. And so my predecessor, Jeremy Berg, liked to refer to us as sociologists of science. And so that's what I'm going to tell you about today. And I have another great colleague who's our executive editor now and runs life sciences for us, Val DeVinson. She refers to what we do as being the people who work backstage. You all, when you publish with us, you're the people that are on the stage. We're the people that are making sure you have your props and that the lights work and that the orchestra sounds good. And as Gil said, I grew up in the theater, so that metaphor really, really works for me quite a lot. But what I want to do today is talk to you about all of these trends that we have in science related to the word open and how it's much more complicated than it sounds, and the implications of it are profound. And those of you who are going to be leading us forward, you're the ones that are going to have to wrestle with all this stuff. And I'm going to pick 3 papers that bring these complications out in different ways and tell you from the point of view of the stage manager how this open thing is going to play out. And of course, there's a lot of good that's going to come out of it and already has, but there are a lot of complicated things to think about. So first, just the slides that I show at the beginning of every talk. And I usually talk to general audiences, so you probably know where to find us on the internet, and you probably know about our 6 journals. The impact factor of science is 63, which is either really impressive or something that you should ignore, depending on how you view impact factors and H indices and all these crazy things that we have. But I can tell you, we had the biggest jump ever this year, and the reason was because of COVID. So one thing you can watch after next year, I think, we'll probably sustain it, but after next year, a whole bunch of these prestigious journals are going to have their impact factors go down when these COVID papers are no longer in our impact factors. And that has never happened before. So this will be a very entertaining sociological experiment for you all to observe, because everyone who's been bragging about their impact factor is suddenly not going to say anything about it because it's going to go down. But of course, it's nothing but just an artifact of what we went through with the pandemic. The science journals, the 5 subscription journals, are different from the journals that I grew up with in the American Chemical Society, in that the people who make all the decisions and handle all the papers for us, it's their full-time job to do that. And for those of you, we're talking about jobs today, it is a really wonderful line of work. And there's a lot of vacancies, just like there are across the labor market. And so it's something for you to think about. You get to work from home, you get to go to conferences, you see all the great science in the world. We have a lot of people who get in that job and stay in it for 30 years because they really have a good time doing it. They're brilliant, brilliant people. Somebody who reads papers for a living is just as interesting to talk to as your faculty or graduate student colleagues at a great department like this. They're just unbelievably powerful scholars. Then we have one large open access journal, Science Advances, and we use academic editors there, like all the ACS journals do, and many of the journals that you're publishing in. The parent journal, Science, is 3 different things. It's a research journal in the back. that I think I had this on the previous slide. Gets 12,000 submissions. We publish 750 of those. The other 11,250 people are mad at me. Yeah, if I ever write a memoir about my time in this job, it'll be entitled, "Sorry, we didn't have better news for you," which is something that I say to a lot of people every day. We're proud of what we publish, and we think it has a lot of impact. The other thing in science is the news department. We have 30 journalists that work for us. They all went to journalism school. They're very traditional journalists, like you see in TV shows about journalists. They go through people's trash and stake out their labs and file public records requests and write edgy stories about things that are going on in science. There's an opening for the director of the Salk Institute, which I think we're probably responsible for. We've exposed a lot of other great things, and there's a lot of careers content that we and all of our competitors are using that I hope you all find interesting, and our journalists all over the world. It's a huge amount of fun to work with these folks. They're editorially independent of me, which means I can't tell them what to cover. I can't tell them not to cover something, but if they screw up, it's my problem. And that's okay, because I really enjoy working with them. And then we have our commentary section, insights. You see the 5 different types of content we have there. This is where scientists talk to each other. So unlike a lot of opinion outlets, our opinion outlet is an outlet for scientists to write opinions for other scientists. And when we think about science as a journal, a lot of people say, well, can you make something for K through 12, or can you make something so that I can convince my climate denying uncle that climate change is real? We don't do that stuff. Scientific American and the Discovery Channel and Weekly Reader, they all, Sesame Street, they do a great job of that stuff. Our magazine is for scientists. So it's for scientists to talk to other scientists. It's news that scientists want to read, and it's research that scientists want to read. How do we impact the public in a few very important ways? The most important is when there's news coverage of our research papers. And we have a whole team dedicated to making sure that the papers get covered and that they're covered accurately. And this is a major issue in science, because when the mainstream media, and we saw this big time in the pandemic, when the mainstream media covers a paper and does it inaccurately, that's what the public reads. So if that's inaccurate, then that gives the public a way to say that we don't know what we're doing, even though our paper is accurate. The news story is inaccurate. That's what the public sees. And this is a major, major challenge. And it's really only science and nature that have teams that work at making sure these things are accurate and go back and get things corrected if they're wrong. That's a very labor intensive thing. And so when people pay for science, the team that handles the earned media coverage is one of the big things that they're paying for. I know your librarian's not here, but that's a plug for them to keep their subscription up with us. Okay, so look, who doesn't like the word open, right? I mean, it sounds good. Let's have this open, that open, everything open. Nobody wants anything closed. People don't like paywalls. People think that all information should be free and democratized. And of course, that sounds good, and there's a lot of great things about it. But we've got, here's the Merriam-Webster's open, having no enclosing or confining barrier accessible on all or nearly all sides. All right, that sounds good, okay? But it's complicated to do. And I'm going to show you some examples where that's true, okay? The first is in what we refer to as open access, which is the idea that the version of record of a paper should be accessible to everyone. even if they don't have a subscription to the journal. The challenge with this is that all these things we do, the journalists, the people who do earned media, the people who work for us full time, managing a Twitter feed with 3 million people on it, you expect all these things to be done by us. We have to pay them for them somehow. And what happened in the early years of this open access idea is that the commercial publishers figured out, hey, this is great. We'll just charge the authors to pay us to publish their paper. And there's a whole bunch of, and you don't see Elsevier and Springer Nature and Wiley, who are the main commercial publishers, yelling and screaming about this open access thing because they love it. Because they don't like negotiating with the librarians for their subscriptions. They can't get very big price increases that way, but they know the authors are really wanting to publish their papers. So if they can charge you $11,000 to publish your paper in Nature and make it open access, and they can get the federal government to tell them you have to pay them that, then not only is, I mean, first of all, they're still getting their subscription fees, at least for the time being. So they're getting that from the librarians. Now they're getting $11,000 from you, and you're at the big old bad mojo factory, University of Michigan, which has enough money to pay the $11,000. But what if you were in ecology and all you had was one NSF grant? What if you were at an HBCU or in a low and middle income country and you couldn't pay $11,000? Well, then you can't publish in Nature. And that's not good for anyone. So this open access sounds good because it means everybody can read it. But it also means that if we're going to subsidize all of these functions, that's going to end up getting charged to grants. And that means only people with a lot of grants are going to be able to publish. And if you need papers to get grants and you don't have any papers yet, how are you going to get a grant if you can't afford to publish? All right. And so there's a lot of inequity baked into this. The other thing is that then the underlying papers are all being given to these commercial entities. So as Gil pointed out, the science journals are nonprofit. We do make a surplus, and we give that surplus to the American Association of the Advancement of Science to do all the important things that they do in science literacy, science diplomacy, all of these science policy, all of these important functions. The commercial publishers make a surplus that they return to their shareholders. And now, so you probably never even noticed this, but if you publish an open access paper, either with an open access journal or by paying an additional fee to a subscription journal, you get what's called a CC-BY license. Okay, now that CC-BY license not only means that the paper is available to lots of people and free, it means anyone can take that paper and change it and do something else with it. So somebody can take your paper, scratch out the part that they disagree with, and publish it again with their own stuff on it. And they don't have to give you anything for it. That's what that CC-BY license does. And most people don't even know this is happening. All right. And so you are, I guess, so a lot of people say, well, I don't like signing that copyright form that gives the copyright of my paper to science or the publisher. But when you get that CC-BY license, you're giving it to every publisher and they can do anything they want to with it. So this open access thing is pretty complicated. It's creating a barrier to publishing in exchange for lowering the barrier to read. And that sounds good. A lot of people weren't paying attention to this. This whole thing got this way, and now we're getting ready to pay a big price for that. Okay, open data, right? So y'all are putting your data on Zenodo and your code on GitHub, all right? And most of the time, you can do that for free. But eventually, as we put more and more data up with every single paper, the cost of storing that and the cost of maintaining that is going to go up and up and up, and eventually, and it's already happening, there are a lot of commercial entities that are going to want you to pay them to host that. And not only are you paying them to host it, but you're giving it to them so that they can use those data, they can resell those data, and they can use them to come to their own scientific conclusions. And, you're creating these behemoths. And eventually, if Elsevier does that, then they'll get good at it and Amazon will buy it and they'll merge it with One Medical. And you can imagine, what's going to happen to all those data that you, with federal dollars, generated. If you have to deposit them, you're basically giving them to Amazon and Elsevier and whoever else for free. All right, open code, same issues there. Who pays? GitHub. How do we decide that it's the actual code? Okay, the author says it's the code, but do we know that? Same thing with open data. We don't go trolling around the internet to make sure that authors didn't put up their code long enough to get the paper, or their data long enough to get the paper published and then take it down. We can't afford to do that. Who's going to do that? So these are all complicated questions related to all this. And I'll take you through 3 little sagas about that. First on open access, I said all this stuff, but here's our policy, which is in this editorial. You notice I'm not an author on this. The way science works, we have a publisher. That's the person who does the business side. I'm responsible for the content. And then Sudip Parikh, who's the CEO of the AAAS, is the executive publisher. So something like our open access policy, that's their deal. I'm supposed to make the best content in the world. They're the ones, and Shirley Malcolm, somebody who works with us on lots of equity and social justice issues, and she's an American treasure for those of us who know her. But in this article, they lay out what our policy is, which is we're not taking money to make papers open access. We're allowing authors to take the accepted version of the manuscript, not the final one where we fixed all the illustrations and put all the links in and all that stuff and copy edit it, the version before that, and deposit it anywhere they want, and eventually it'll be on PMC. And you can say, wow, you're giving away a lot there. Well, we are. But the alternative is to charge for it and to charge direct costs into grants. And we don't think that's good for science. So we're hoping that the librarians will continue to buy our subscriptions and that we can do this as a way of satisfying the demand that people have for open access without double dipping like the commercial publishers are doing. And one thing about the paywalls, whenever you see somebody on social media complaining about a paywall on a paper, it's almost always somebody who works at a university that has a license. They're basically just saying they can't be inconvenienced to log on to their VPN to read the paper. And I'm sorry, if I'm an author in a low income country and I'm going to be excluded from publishing because someone at the University of Michigan thinks it's too inconvenient to go on their VPN, that doesn't hold together as far as I'm concerned. So, I know it's a drag to log onto the VPN if you're at home, but tell your librarian to make the VPN better, Okay, so, I usually give these talks for very general audiences. I'm bringing the coals to Newcastle with this slide, but I think you're probably all familiar with the work in applying artificial intelligence to protein structure prediction. I know there were groups here that have done a great job with this. I'm going to talk mainly here about the Google group and David Baker's group. But this is how I explain protein folding to a much more general audience than this. But there's an important thing here because the solution to this required 2 things that relate to open access, open code and open data. One is that the PDB had to have been built because we wouldn't have had this protein structure prediction without somebody having the vision to build the PDB whenever it was 30, 40 years ago. And the other is, what's that? Even longer? Yeah. It's no good if the code isn't available to everyone. because then, it's just Google winning a contest. It's not actually taking protein structure to everybody who deserves a chance to use it. So, again, this is how I explain things, but this is also a plug. This is the drug I developed. This is the crystal structure of it. So I always like to show this. But of course, this is the reason why this is one of the reasons why we need accurate structures so that we can do this without having to get a crystal. And I like to tell audiences that this all started with myoglobin. Brian was correctly saying lysozyme was also important, but lysozyme doesn't have a metal in it, so I like to show this one since I'm a bioinorganic chemist. But this is where things started, and here's here's the first 50 years of this. We went from Perutz and Kendrew winning the Nobel Prize for myoglobin at 6 angstroms in 1962, and then another Nobel Prize for cryo-EM in 2017, where we got to the point where we didn't have to crystallize the protein in order to get these structures. But then we get to the state of the art in 2012. I didn't plan this, but I guess 2012, CASP started the same year you started your department. Is that correct?
Started earlier, but that's when we started winning. Right?
Yeah, okay, that's when you started winning, even better. Yeah. CASP started in 94? Yeah. Wow, okay. So this is some paper I found with a bunch of the CASP structures from 2012. These are relatively small proteins, but everybody was still really excited about this. I was in an administrative fog at the time this happened, so I didn't learn about this until more recently. And like I said, this is from my predecessor, Jeremy Berg, who points out eloquently at the time that all of this kind of came to this conclusion that, we wouldn't be where we are if it hadn't been for the visionaries who set up the PDB. And, he goes through, this is the most important paragraph of all this, that the cultural shifts were often, controversial, but it is hard to argue that, the structures wouldn't have happened if it weren't for the PDB, and the structural prediction problem has been solved. It's hiding in the PDB, he says, and I think that's absolutely right. And so on the good side of open is the PDB and the visionaries that set it up and figured out how to host all the data and made it so that we can download the viewer and look at all the structures. And, people who came along later just take all that for granted, that it's straightforward to do. I'm kind of in the middle on that. We certainly, in the 80s, we had plenty of protein structures, but you couldn't just get on a PC and play with them till much, much later than that. So all of this has come along during my time in science.
My last PDB looked like phone books, and we had mag tapes. And we wanted
there you go. Right.
There was no real way. There was no internet.
Yeah. So once the PDB came to the internet, it was pretty amazing. All right. So you all know this. DeepMind started winning CASP. And of course, they got a huge group at Google that they applied to this. Of course, I haven't this animated in the PowerPoint version of this talk, but I knew you were PC people, so I sent PDF for my slides. So, but you've all seen these movies a million times before anyway. Google's winning CASP. They are obviously closing in on the answer to this. But initially, their algorithm was not widely available. So they get, right after the latest CASP that they won. Of course, we know there's going to be a big paper on this. They've always been nature people on their protein stuff, but of course we had to make a run at trying to get the paper. So we talked to Demis and told him how great it was that he'd accomplished all this stuff, how it would look great on the cover of Science, and everything else we could think of to tell him. But and we made a decent pitch, but he decided to go to Nature. But in those conversations, it came up, well, what are you going to do with the code? If we publish this, will everybody be able to to, go somewhere and do their own predictions, or how's this going to work? Because up until that time, that code was not widely available. And, we probably would have taken the paper if with some kind of agreement that we probably wouldn't have been thrilled about the access, because it was a big enough paper and we wanted it. But, we were worried about that, and we were debating that. So he goes to Nature. We're licking our wounds and saying, oh, well, we tried to get it. Then we get a phone call from David Baker, who says that this brilliant postdoc of his, Dr. Bike, has figured out how to do this a lot more inexpensively, and that they want to publish the paper with us, and they're going to make the code freely available. So we thought that was pretty awesome. We wanted to get the paper out before Nature did. We reviewed it got it reviewed very quickly. The reviewers got all over their algorithm and tested it. And I don't know, somehow Nature found out we were up to this. And so these two papers ended up getting published on the same day. And lo and behold, Google decided to make their code much more freely available than we were expecting. So this is a great story. We're proud of the part that we played in catalyzing this. And now you all can decide whether AlphaFold or Rosetta Fold is better, and you can pit them against each other, and that's the way it should be. So this is open code doing for good, all right? And this all played out really great. And we made it breakthrough of the year. And I wrote a glowing editorial, which I always write for the breakthrough of the year, saying the protein folding problem was solved. And a whole bunch of biophysical chemists got very mad at me because, of course, they don't see AI structure prediction as solving the protein folding problem. They just see it as a way to get the structure. So I'll let you debate that with them as well. But I love it if you argue with them and end up generating some great papers that I can publish. Very similar to that, we started getting papers on RNA structure prediction. There's a lot of commercial activity in this space. I have to have a disclaimer because I'm actually a partner of a venture capital firm that owns part of Ribometrics, but Ribometrics has got a lot of great competitors. And RNA targeting by small molecules is super hot in the biotech space right now. And using these same principles to get RNA structure may get us more drugs than on the protein side, who knows? But that's exciting too. All right, so that's the protein folding story. Now let's go to another even hotter topic, which is where COVID comes from. And I'll just kind of take you through the timeline of how things have evolved. So, and we played a very important role in this throughout the time that this has been going. I'm making sure I've got plenty of time before Brian yells at me. So in the early days of COVID, there wasn't a whole lot of discussion about where COVID came from. There were some people saying, oh, the Chinese must have engineered this, or it was engineered in Chapel Hill, but there was a lot of people thought it came from the wet market in Wuhan, and not a lot of discussion. Well, then there's pressure from politicians, and eventually the WHO sends a team to Wuhan to look at this, and they wrote a report that said that it almost certainly came from an animal. We call that zoonosis. And that it was highly unlikely that it originated in a laboratory, which turned out to be correct. But a lot of people felt, and I agree, that if we were going to defend that conclusion to all the conspiracy theorists and politicians who were trying to tell us that was wrong, that we needed to do a better job of ruling out the lab leak hypothesis more rigorously. So we published a letter, which a lot of people objected to. But like I said, when we get in trouble, that's my decision. So it was ultimately my decision to publish the letter from Jesse Bloom, who's in the upper right-hand corner of that picture. who's a virologist at Stanford, probably the most prominent virologist who still thinks that the lab leak theory is viable, and 14 or 15 other scientists who all felt that there should be a more rigorous investigation of the lab leak. Now, a lot of those people who signed it were not necessarily, and me in terms of deciding to publish it, were not necessarily people who thought that the lab leak was more likely or even somewhat likely, but who felt that the lab leak should be more rigorously ruled out if we could, so that we didn't have this lingering debate about all this. But a lot of people criticized us as saying that by publishing that letter, we were fueling the conspiracy theorists. So looking back on it, we're pretty happy we published the letter, because a lot of the authors who are on there did start digging into this and came much, much further to the conclusion that zoonosis is so much more likely, as that the lab leak is almost completely ruled out. And one of those is Mike Warraby, who's in the middle on the top of that, who's professor at Arizona, and I'm going to describe his paper to you. So Mike signed the letter saying we needed more investigation, and then went and did the investigation. In the lower right is Alina Chan, who's one of the strongest proponents of the lab leak. In my opinion, mostly on behavioral evidence, not scientific evidence. And then that's Lin-Fa Wang, who's a hardcore zoonosis guy. And in the upper left is John Cohen, who is our journalist who covers all of this. And if you haven't seen it, I highly recommend the HBO documentary, How to Survive a Pandemic, which is all about John Cohen going and finding out a lot of this stuff. And he's a great and entertaining journalist in every respect. And he hosted this debate that we had about this. Then we started publishing papers about this, some mostly perspectives at the beginning before there were data. Eventually, we published this perspective from Mike Warraby himself. This is a map of the early cases of COVID. And you can see here are the two labs, here's the market, and the cases are mostly around the market. You can just tell that with your eyes. There are a few cases down by the lab, but not nearly as many as there are up here. So this was kind of the first stake in the ground here. But eventually, Mike's group did a much more rigorous study of all this, which we published about, 8 weeks ago. And these papers, so here's the first open access part of this. These papers were all in preprints, all right? Everybody here knows what preprints are, I'm sure. Science is very much in favor of preprints, because we like it when the experts can go ahead and start talking about their data. And we don't think it takes anything away from us when we publish the paper, because if something's a preprint, even if it gets covered in the news, when we publish it, it's way bigger, because we have all these teams that make sure that our papers are heavily covered, and that was certainly the case with these. So these were in preprints. They were controversial, and the lab leak crowd was all over social media, taking them apart. But we had them reviewed. We studied them very, very carefully, because we knew they're going to be read on the floor of Congress. And whenever that happens, it's all hands on deck. So this figure from the paper, these are the cases that the World Health Organization has in their data from December of 2019. And the orange cases are ones where there was a known link to the market. And the blue cases are ones where there was not a known link to the market. And as you can see, just with your eye, they're pretty well aggregated around the market, even the ones that don't have a known link. And of course, the other open part of this is that these data were on the internet, and Warraby was able to get them to analyze them. And here's, these are pretty simple calculations to do, but if you calculate the epicenter of all these cases centered in the market, if you take the linked cases out, you still get a pattern that's aggregated around the market. And if you then take even more data, everything, not just the WHO data, but data from lots of sources, those also center on the market. And these are the centers of all of those. And over here, that's the center of the Wuhan population. So if it was totally random, you'd expect it to be over here. And all these different subsets are all of these points over here. And what's really important about this, which I'll say more about in a second, is that there's another paper that the same group published showing that there are two distinct genetic lineages of these early cases. And both of those lineages are also centered on the market, okay? This is a great kind of open data thing. These are check-ins of different sites on social media in Wuhan, right? So these are supermarkets, shopping malls. This is how many people, it's a big city. These are how many people checked into the biggest one. You see buildings, hospitals, schools, markets. All the way down here is the Hunan market. So the Hunan market is not the center because there are a lot of people going there compared to other sites where the virus could have spread more easily. And then here's a figure that shows a map of the market itself. This is where the animals are kept. And all these little squares are where there are human cases. So almost all of them on the same side of the market where the animals are. There are a few over here on the other side. So all of this pointing pretty strongly to the market being the epicenter, and that the animals are the source of the virus. And it's hard for us to understand, but there are millions and millions of live animals being traded in these environments. And so the flow of animals through the wildlife trade in China is just absolutely enormous. And that's a very conspicuous fact in this. And the other thing that's important is that this is how we got SARS-1. So even if you were to discount all this, you'd have to say that there's some reason why we got SARS-1 from zoonosis, where we actually found the animal that started it, and somehow SARS-2 didn't form that way. Okay, so like I said, the same group had a paper that we published together on the molecular epidemiology of this. There are two distinct lineages. And the first was introduced around November 18th, 2019, and the second was probably a couple of weeks later, right? So now, go back to the map and ask yourself how you could get this map given these facts. Well, you'd have to have somebody at the lab who got infected by an animal or ridiculously by an engineered virus that they made, or I don't know. I don't get how that part works. But let's say they had an animal that they took to the lab that had COVID, they got infected. They'd have to go to the market, infect an animal or a human, and go back to the lab and not infect anybody else. And 2 weeks later, somebody with a different strain would have to go to the market, infect another animal, and go back and not infect anyone. Okay, so does that rigorously rule out? Is it completely ruled out, the lab leak? Well, no, because we're all scientists and we know we don't ever completely, no rate constants are zero. and every finding is subject to more investigation. Do I think it's likely that we'll get a paper about the lab leak that we'll want to review that will blow this up? I think it's extremely unlikely. Would we review it if it was credible? You better believe it. You think this is big. Just imagine if we got the paper that proved that the lab leak was right. But of course, I think it'd have to be very rigorously reviewed. And I think it's very unlikely that that's going to happen. So, you know, a lot of the people who shoot at this say, well, this is all groupthink because the scientists want to make themselves look good and don't want to make it look like they're sloppy in the lab or whatever. I would definitely review the paper if I got a credible paper about the lab leak. I promise you. So that, you know, that's what's great about science. The fact that we love proving each other wrong is a major protection device that we have that the public doesn't appreciate. All right, so now the thing about these papers being in preprints is, of course, all the lab leak folks read the preprints very, very carefully. And when we published them, they were toned down compared to what the authors sent us. This is what happens with every paper. We tone down every paper. We don't allow the word first. We don't allow the word breakthrough. We don't allow the word novel. We don't allow the word, you know, we don't let people hype papers because if they're in science, they're already hyped. They don't need to be hyped anymore. Am I going on Star Tours? Yeah, okay. All right. So sorry, I live in Orlando. I hear that. That's the Star Tours sound. So we don't need to hype papers. So we tone them all down. And you know, the conspiracy theorists got, you know, they had the word dispositive in there. Okay, we said, okay, guys, dispositive is a little over the top. Because of course, you know, we wouldn't want to tell anybody because, you know, the first thing's going to happen. Eventually, there'll be a hearing in Congress. Mike Warby will have to go there. Rand Paul will say, is there a chance that have you completely ruled out the lab leak? And he'll have to say, well, no, because you could have this sequence of very improbable events that we can't completely rule out. So and then Rand Paul would say, oh, well, you said it was dispositive. We didn't want that to happen. So we changed things like, we changed it to stuff like strongly suggests, okay, and highly supportive of zoonosis and stuff like that. And the conspiracy theorists think that the reviewers made us do that because they weren't persuaded by the data. They weren't. I and Caroline Ash and Val DeVinson, who worked with me on these papers, we're the ones that made those changes. And we know these papers were going to be highly scrutinized. We went through every single word of them. It wasn't because the reviewers weren't convinced. So that's another open access part because the preprints allowed the comparison with the published version. And the open data part is that all these data were accessible. And the political part is that a lot of people said that, well, and this gets to a lot of stuff Brian was talking about anti-Asian bias in America, that you know, the people who are the liberals who are pushing the zoonosis are doing what the Chinese government wants. Well, at least as far as science is concerned, we love Chinese scientists. We don't necessarily love the Chinese government any more than we love the American government. And the Chinese government is pushing the theory that is just as improbable, if not more, that somehow the virus was transported into China and then and it didn't come from an animal that was raised in China. And we have a big news story about that John Cohen wrote called Anywhere But Here. That's the Chinese government's slogan about this, which I think is just as wrong. So the conspiracy theorists who say that we're pushing the zoonosis because, you know, we want more Chinese papers in our journal, or because we're you know, we're too whatever they think we are, cooperative. You know, the Chinese government, George Gao, who's the head of the Chinese CDC, is cranky with us that we publish these papers just as much as Rand Paul is. So, you know, the science has to ultimately be the thing, not not the political part. But it's certainly not true that that this is some kind of scheme to reward the Chinese government by sorting this out. And I wrote a very tough piece at the beginning of all this about the failure of China to disclose more information about the pandemic early on. Okay, one last thing, example. Looks like I'm going to be perfect because this one's short. About open data. So these big companies have a lot of amazing data. I mean, you could tell that from Aaron's talk just now. And I'm sure Dan has got some nice stuff on his servers that the world would like to see, and that could drive a lot of great science. Yes, but you know, the Nordstrom and Strata want to charge people to look at those data. And there's a lot of this coming now in social science because Facebook and LinkedIn are sitting on the greatest treasures in the history of social science data. And the scientific world could do two things about this. We could say, we're not going to publish all of that because it came from proprietary sources, or we could try to partner with them to come up with ways to use their data to do important things in science. And so we've started venturing into this. So this is our first foray, which was just from last week. There's a big theory in social science called the strength of weak ties. And for all the grad students who are going to be looking for jobs, what this says is, you know, if you met me today and you friended me on LinkedIn and then you asked me to connect you to somebody to help you get a job, that's more likely to work than it is for somebody you've known your whole life. That weak ties are actually more useful than strong ties. And there are lots of ways this plays out in social science. This theory's been around for a long time, tested many times, but there's no better way to test it than with the data that LinkedIn has. Because everybody's on LinkedIn because they want to get a job. So it's very easy to measure the success. And LinkedIn has incredible data on the strength of, you know, the connections that people have to each other. So LinkedIn made a bunch of these data available to some outstanding social scientists. They sent us a paper, we reviewed it. We worried a lot about the terms. So you can see what we agreed to here. All original analysis code, aggregated data, and annotated code sufficient to replicate all correlation regressions are available here. We know this, we know this link works. Individual level data supporting this study are not public available due to user privacy and other legal restrictions. Researchers seeking replication should submit a request to LinkedIn, and you know, they get to decide whether they give you the data or not. All right, is that perfect? No. Would I've preferred it if LinkedIn just said, here's all the data that we pulled this from? Sure. They weren't going to do that. So we had to make a decision whether to agree to this or not, and whether to publish this paper. We got a bunch more of these in the pipeline, and they're all blockbuster papers. This paper was all over the news, and we think this is maybe not even the most significant one that we've seen. But still proud to publish it, and it was great, excellent science paper and big deal in social science. You know, we're getting some serious snark about this. Some of it we deserve. You know, the New York Times wrote a tough piece. If you're in the data science world, as you all are, I actually encourage you to go read this, even though they said some things about me, not me personally, but about our journal that I didn't love. But you know, they framed the problem here. So guess what? This study came out great for LinkedIn. Okay, because it shows that if you're on, that LinkedIn is a great way to find a job because that's how you can get the most weak ties. All right. So how do we know that? So if the data hadn't shown that, would LinkedIn have given it to us? Probably not. Yeah. Not probably not. They wouldn't have. And of course, then there's another thing, which is this experiment was done on millions of people who didn't know that this experiment was being done. Now, you know, when you create your LinkedIn account, you click on something that says, I agree that you didn't read. That gives them the ability to do this. So, you know, that's bringing awareness to that is good, I guess. And so you can see some researchers who are kind of activists in this space. And this is a guy we love, Dbtl Cycle, who works in this space, but he was not happy with us for doing all this stuff. And, you know, I see where he's coming from. But I didn't have the option. So, you know, he's saying we should have just not published this. Well, it's a very important theory in social science. We vetted all the terms with really experienced social science researchers. They agreed we should go ahead with it. We peer reviewed the peer review in a sense when we did that. And, you know, it's a high impact paper. So we decided to publish it. Now, I'm glad we saw all this because we got a bunch more in the pipeline. And, you know, watching how this played out helped us think about how we could do this better in the future and what we might get out of it, or how we might, you know, lower some of these concerns. But these are the questions we're all going to be wrestling with, because if LinkedIn's not showing their data, let's say Dan decides he's going to let you write a paper about his stuff, and it's awesome and proves a big thing about cancer, and we want to publish it. How are we going to go about deciding whether that's, you know, we've adequately controlled for all this stuff? That's a work in progress. And so I think the point of this whole talk when it comes to open access, open code, and open data is, as I said, the devil's in the details. It sounds good to have all this stuff open, but you all, as you go forward in your careers, are going to be spending, and even right now, are going to be spending a lot of time peeling this onion, because every time you think you've figured out every way to make the code available, there's another thing that comes along. Or, you know, I have a really cool job in so many ways. And, you know, sometimes I have to decide things like, well, we got this rock out of the bottom of the middle of the ocean. And here's what we concluded from it about the evolution of the Earth. But we can't make this rock available to everybody. So what do we do about that? Or, you know, we got these fossils and we can't make them available because they came from this indigenous land and we can't take it from there. So we can't put it in a repository where other scientists can come observe it because we'd be, you know, appropriating it. And now we got all the same things with data and the code and the papers. Who can change it? Who verifies it's right? Who pays to store it? And who decides that enough is enough in terms of what's open and what isn't? So those are the challenges I leave you all with. I'm happy to hear your thoughts about this. This is something that's going to continue to evolve. And I'll skip that since Brian's about to yell at me. And here's the team that brings you Science Magazine every week. Like I said, these are some of the smartest people I've ever worked with, and I've been in a lot of great academic departments and a lot of great schools. But these are brilliant scholars who read papers for a living, and they have an unbelievable ability to determine when they're correct and when we should publish them. And you know, I think of them as my faculty colleagues in every respect. So thank you so much, Gil. It's an honor to be the Omen Lecturer, and congratulations to your department on 10 years of great success.
Sure. That's okay. Yeah. You know, we wanted to have a panel discussion because we really wanted to explore some of these things with you. Sure. And the map you want to put down. Okay. I think we've got plenty of time to get started.
So I because it actually has to do with Aaron and your talks. Because if I have to moderate this, I need people to moderate. So June, yeah, June is down here too. That's- All right, so while everybody's getting ready and they're doing something around here, I actually want to say congratulations to Brian and Gil on the 10th anniversary. I'm actually probably the only person that does not have an appointment in the department, but I actually have published, I think I counted 8 people that I published with here. And I want to thank Brian and Gil for all the support they've given me in developing the programs that we've been doing. So I actually, I'm glad I got a chance to say this in public. All right, so I'm going to start off with one question, and then I want to open it up a little bit to do it. So, a lot of things, and June, I'm glad you're here too, because June is one of the people I published with. It's leadership. All of you are leaders in your own way, and but I'm wondering how you learn to be a leader. Because this is a really important thing to really get things done, especially if you're going to continue to lead or want to lead groups and not working by yourself. And so I wondered if you could address what the ups and downs, and is this natural? Do you learn it? And how does this evolve? So I think Holden, you obviously have risen to more leadership positions, and I'm going to get your impression first, but then I actually want to bounce it out to actually I'm going to talk to Aaron too, because
Yeah, well, I've talked enough, so I'll just say something that I already said to the students, which is the most important thing about leadership is that positive reinforcement works and negative reinforcement really doesn't. And and I learned that from Harry Gray, my research advisor, who's the world's most positive person. He's a wild extrovert, and all he does is just tell you what a great job you're doing. And then occasionally he tells people to leave graduate school. But he doesn't do anything in between, because when you give people tough feedback, no matter what HR tells you, it actually doesn't work. It makes them perform worse. And I knew that when I was running my lab. I struggled to learn that again when I got into, the spotlight more in leadership, but then I later figured out, that was right all along. And so, especially in this labor market where people are getting out of the workforce, being a source of positive encouragement is the leader's number one job.
Thank you. So yeah, I agree. It's very important, and I really also want to echo something that Dan said. To me, it all starts with feeling passionate about what you do, because without that, I don't know how you can sort of lead anyone. And so I think also it is about creating an environment in which people feel comfortable sort of expressing their opinions, and I think it sort of ultimately leads to great results. So just
thanks. So I'm going to pin you down, Aaron, because you now are a leader, and so you had a, I don't want to say subservient position at the clothing manufacturer, but so how did you grow into this? Because I know you very well, but it's and I think I know, but
Sure. So just like we were saying, we talked about this a little bit at the student lunch, and for me, that was a really natural progression, going from being a technical contributor to then ultimately kind of setting the strategy and agenda, and then recruiting and building a team. But I think in terms of just what's the important shift that has to happen for you to do that, I think one is the recognition that you got to that space because you were excellent technically, in my case, for example, but there's a shift that has to happen for you to continue sort of being excellent as a leader. And I think one of the core pieces of that is really the change of mindset and the recognition that it's not about you anymore and that your success is your team's success. So instead of thinking about my technical contributions or, making sure that my name is on the paper, it's more about what's in my team's way, what is the barrier for their success, and then my job, or where do they want to go in their career, and then my role becomes enabling that to happen, or making sure that they have the right types of work to grow the skill that they're trying to grow and to get to unlock the next stage of their career. So it's really just a mindset shift, that the way that you succeed when you're moving up to a leadership role is not your stellar work and the spotlight on you. It's your team's success, and then of course you get to share in that as well. But yeah, it's just a difference in focus.
Okay, thanks, Aaron.
Sure.
Yep.
And there were a lot of different things that I think we could talk about. One of them I'd like to start with is a little bit more in-depth analysis of this page charts thing. One of the things that you said that bristled me a little bit was that the mighty rich University of Michigan could easily afford these $11,000 page charges, and the poor guys out in the hinterlands couldn't. Well, the assistant professors here couldn't do it either. And, it's going to create some kind of uneven playing field. So you have the forces of Elsevier or Springer-Verlag over here. You have the forces of bioRxiv and medRxiv over here. You have a unique situation with the science journals. You have the libraries in transition still towards a fully digital, remote, maybe even non-existent state. I mean, what's really happening here, and what needs to happen, and how can all of our experiences, and I want to hear from that, kind of inform this, because we're running into a little bit of a train wreck, I think.
Yeah, we certainly think so. And yeah, of course, the assistant professors first starting out who are spending their startup packages can't afford $11,000 either. But it's also true that the provost of the University of Michigan could fix that problem. But the provost of Morgan State can't. Or, I was thinking, Western Michigan. And so, so yes, I think it's a train wreck. And just think about it, if every single paper becomes an APC paper, and the average is, let's say, $3,000 or $5,000 per paper, then every lab's got to have at least $50,000 just for the APCs. And if you're a big old cancer lab, okay, that's probably doable. But if you got one NSF grant, that's a third of it. So yeah, the scary thing is, if all of it goes that way, and I don't think it will, but if it did, then Elsevier can raise the price without having to negotiate with the librarians. And that's why they want to do it. That's why you don't hear them complaining about it. I mean, there are a lot of, these people who are these OA zealots on social media the day that the OSTP said this change was coming, who said, whoa, I wonder if Elsevier is okay. And I'm like, is Elsevier okay? They're laughing all the way to the bank. So yeah, this is a complicated problem. I'd like to hear from my other panelists, though.
You're publishing 10 papers a year. I mean, it's just big. It's a big impact, and it likely will come out of some kind of non-existent discretionary fund that we don't. I think you have too much faith in the provost, especially over here at the medical school.
I'm sympathetic to provost.
I know you are. Indika, go ahead.
I don't have any. So I publish in like IEEE instead of.
Yeah, good. Put that microphone.
Yeah, and so I train as a mathematician. So in mathematics, like great papers sometimes in open, like just put into archive and leave it there. And like 2 weeks ago, one of my paper accepted to Nature Communications. So the bill came, it was like $6,000.
That's what it is.
I was a little bit shocked, but I have in my lab, we have funds to pay that. But then this week, another paper got accepted to IEEE instead of electrical engineers. It's free. I don't want to give anything. So, and I'm coming from a third world country. I'm from Sri Lanka. And I really like it to be open, like friends in my country can read papers, right? It's, we need to adapt somehow. I don't know a solution, and we need to adapt somehow how to make it open source.
So I want to ask you something. So, you know, these publishers get a lot of free labor. Do the scientists have any power in this to change this the cost structure, or you know, demand that if you review, you know, X number of papers, you get free paper? I mean, we have the power to withhold our publications, but we need to publish. So do they have us? Or is there something?
Well, certainly in certain fields. I mean, you know, that's why what we're doing is we're continuing to charge the libraries for our licenses. University of Michigan's the biggest, so they pay the most. It's progressively priced. So your library is paying us about $40,000 to get our 5 subscription journals. Sounds like a lot, but you're paying Elsevier $10 million for all of Elsevier's journals. So we're banking on the fact that when your librarian has to cut their budget, we're not really on their radar. And so we're just going to start giving away a whole lot more content, so that, you know, if you're in Sri Lanka, you can't get past the paywall on our site, but you could read the paper on PMC. That's right. And you know, we're counting on the community sticking with us on this. And so, you know, there's one thing you can do, which is, if journals try to do this, which we think is a more honorable thing than what the others are doing, you know, buck up your librarian if they say, well, wait, these papers are on PMC, why am I paying for this? Well, you're paying for it so that we don't have to charge you way more than $10,000 per paper. So that's one thing you can do. And the other thing you can do is try to help more people be aware of the fact that, you know, these journals have different models. I mean, a lot of people say to me all the time, oh, well, we think of Cell Science and Nature as all the same. And in many ways they are. But if you publish with us, you're helping the American Association for the Advancement of Science. And if you're publishing with Cell or Nature, you're publishing the shareholders of Springer Nature or Elsevier. So, and I think most people aren't even aware of that.
What do you think? I'll let Chuck lead. Do you want me to open a different topic?
Absolutely.
Okay. I'll build on a different topic, because Holden, you started early in your talk about how scientific journal editors are scientists who study the behavior of scientists. And then you must have observed the different types of authors, their style of proving the way they build a case. And then you select 750 as published. How do you build the expertise among your editorial staff and among reviewers, or how to give feedback to those rejected papers so that there is the collective increase of expertise?
Yeah, that's a super sharp question. And the biggest, you know, thing that I wish I could figure out is what you raised at the end, which is, how could we give more feedback to the people who don't quite make it? Because that's a lost opportunity. You know, so of the we publish 750, we review 1,500. So the other 750 that doesn't get published are getting some feedback, but particularly, you know, the assistant professors or the people from other schools that don't have as many colleagues who've published with folks like us, you know, it would be great if they were getting more feedback, but from a cost perspective, I mean, when you got 12,000 papers to chug through, you just got to get to the next one on your queue as fast as you can. And so asking my folks to do that isn't in the cards. So if anybody has any ideas about how to do that, I'm all ears. That would be really, really important. As far as the editors, We've got a lot of editors that have been around a long time, much longer than I have. I learned everything I know about publishing from them. And so we just have a tradition of bringing young people in, having them stay in the organization for a good while, and them training the next generation. And, again, I think the fact that we're nonprofit and we're not enormous means that we have more of that than our commercial competitors do.
Thank you. So I want to encourage the audience. Do you have questions? Comments? Please.
Holden, great to have you here. Thank you for the great talk. As we're moving into more of a big data world, like the LinkedIn paper that you described, how do you ensure that you have the domain expert review, but also the data science review? I know in a couple of times years ago, I reviewed papers that I actually wasn't so much the domain expert, but I'm a data thinker, and dug in and found common flaws in data, and that led to paper rejections, and have found those sometimes after the fact of papers being published. How do you balance those two? And when associations are being identified that they couldn't have arisen by chance, that really good rigorous data science and statistics is going into these types of studies?
Yeah, so it's not perfect for all the reasons that you just said. And one of the things I think is really important is for everybody to recognize that it doesn't end when the paper is accepted. It doesn't end when the paper is published. We do about 5 retractions a year. People ask me all the time, oh, aren't you embarrassed to have 5 retractions? No, I'd be embarrassed if I didn't have 5 retractions, because it would mean that, A, we're not taking enough risks on some of these things to push the boundaries. And B, we don't have a robust process for following up on papers when there's a challenge. So I think, one of the biggest things we need in academia is to try to lower the stigma of having your paper corrected or even having it retracted, because that's just part of the scientific process. And we've got a major sociological problem with that. And we're working hard to change that, but boy, it's tough. But we had a science translational medicine paper a month ago. Some image sleuths went on and found a bunch of screwed up Western blots. If anybody can think of a way to do Western blots without taking a photograph, that would be a really good thing for science. But some photoshopped Western blots. The lead author didn't know about it. We told him about it. As soon as he saw it, he said, retract the paper. If every story played out that way, the scientific record would be way stronger than it is now. And so we need to change the behavior of scientists to get them more comfortable with this.
I want to go a little bit more into the other data openness and the one-way streetness of it. So when the Human Genome Project started, it was one of the first projects where all the data were basically released to the public the day it was generated, and that was part of the agreement. But then a company decided, oh, we want to do our own Human Genome Project, had access to the public data. And as far as I know, there was a huge debate that people have feelings about of whether they really got the human genome separately or whether they just did get some of the public data. And now fast forward, we have a database of huge number of genetic data in dbGaP that we are obligated as scientists to submit if you're NIH funded. But the Chinese government has decided that their own data is property of the Chinese government. So there is I've worked in China, and I love the access, but on the other hand, the access is a one-way street. They have access to dbGaP, but we don't have access to their data. And there may be many other examples like that. How do we deal with the fact that some people have access to everything when it's open, but there isn't a reverse direction?
Great, that could have been part of my talk, but I'm going to let my colleagues up here respond, and then I'll say something if needed. June or?
Well,
you already commented on the political crosswinds and different nations. In addition to that, not only the geopolitical concerns, there is also a cultural difference. We sometimes wonder why climate science and vaccination is such a hot topic here. And in China, it might be genetically modified food. In Europe, it's animal rights and nuclear energy. So it's hard to really study the comings and goings of these concerns in society, but that's where scientists are the true citizens who are without borders, right? If we talk in our community, we become, again, world citizens to help breaking some of these barriers. I guess that's my comment.
I have no comments about that. I teach mathematics of data, and sometimes I wonder the integrity of data. Sometimes there are a lot of data. How much I trust the data, right? And I have no comments. I have no comments. Thanks.
I was going to take it in a slightly different direction, if that's okay. So when you, in your talk, when you talked about the origins of COVID, I think you touched on the sort of, rise of various conspiracy theories and things like that. And so what I'm kind of wondering is, what do you think is a way to, as scientists, I mean, and sort of, being the editor of, science journals, so do we have a bigger role to play in combating this? Because I kind of, I was, I grew up in the Soviet Union, so I kind of know firsthand where kind of, disinformation and propaganda can lead. And so I think we should be very concerned about that. And how do we combat that?
Yeah, so you're teeing up something that is I've been putting a lot of emphasis on, and hopefully we'll do more. And that is, and I'm doing my first outing at GW on this. I mean, I think everybody who gets an undergraduate degree in science, and certainly anybody who goes on for PhD, needs to learn the history of science and the politics behind how a lot of these things played out. Because all of these patterns that we've seen, and definitely in other countries where, June's absolutely right, about the different issues that play out in different places. Politicians thrive on doubt. Doubt is the most useful political thing in the world and it's way more useful than certainty, right? So, my political views are no secret. So, why did Donald Trump not do anything about, really investigating Hillary Clinton's emails? Well, because the answer wouldn't be nearly as useful as the fact that he can still use it today, because he sowed enough doubt about those emails that it's still useful to him, right? And the same thing is true about the lab leak or about the vaccine causing myocarditis or, all these things that we've debunked. The politicians don't want those things debunked. And most scientists are not very aware of that. And this pattern has been going on for at least 100 years, if not more. I mean, really more. Galileo went through exactly the same thing. And, I'm a huge admirer of Francis Collins, but he stepped in it when he said, somebody asked him, what could you have done better in the pandemic? He said, I wish we knew where hesitancy comes from. Well, every historian of science and political scientist was, what's that emoji where you're going like this? With that, because that kind of thing's been worked out for years. But, I didn't really know all that stuff when I was going through my education. I didn't really even start thinking about that until I went into administration or even after. And so we're really shortchanging the scientists that we're not preparing to understand, and the open access thing's another great thing. Because, what happens? People saying they're doing something progressive that ends up harming the people who are marginalized. I mean, that's what this APC deal is. Elsevier is saying, look, we're open, you know, and meanwhile, they're closing out people from publishing. And that's no different from redlining or, you know, a million other sociological things that we've had in the world. And the fact that scientists aren't required to have an appreciation for this, I think is hurting us, and we need to correct that.
Great.
Hi, can you hear me?
Yeah.
Okay. I'm a graduate student in Dr. Arul Chennaiyan and Marchand Cieslak Lab, and I have a recommendation and a very general question. So a very naive recommendation for retraction papers perhaps can be, it's a bit controversial as well, that maybe hold the reviewers accountable as well. Like if you retract the papers, the reviewers also sort of come up because they're supposed to be accountable for that paper as well. This is just a naive recommendation. And the question that I had was regarding the idea of integration and moving forward with data. So like Dr. Maureen's presentation, we see that different waves of bioinformatics coming up. So there's microarray, there's single cell now. So as you see, single cell data and spatial transcriptomics are more or less done in every lab, right? But there's a lot of microarray data that is untapped, right? And that could hold some potential into new discoveries. But people are moving towards single cell and spatial transcriptomics, so they're not really focusing on that. So how can you, or as scientists or something that, or someone who can sort of influence media, can encourage researchers to tap into those resources which are right now unlooked at, or they're not as important right now?
I guess one reaction is the research is funded in this R1, U1 scale. So there is very little add-on for integration margin to make it usable, to make it overlappable with the next R1. So I guess that's really a decision, not only for researchers, but also for societies and government and citizens to figure out how to create that add-on. And then on the up, the beginning of the game, there is also the cohort development, right? A lot of NIH now fund discovery projects, but not for biobanking and resource building. So we have like a sparsity of attention to the beginning of the game and the end of the game. That's my comment.
Maybe so what you're saying is that new technology comes, then you have better understanding. Then you're saying that like microarray, a lot of is that data is I think technology developing more and more, we can see more detail about the problem you're studying. But I think also you can use this previous data to make good questions, not to answer, right? There is a project I am working with Dr. Oman as well as Dr. Lee Hartwell. We are thinking to create like a catalog, not just to answer, make questions, good questions from existing data, right? But I believing in technology, right? And new technology comes, you have more detailed understanding of biological systems, right? But integrate both the previous data, new data, and think mathematically to how to integrate.
Dr. Kretzler, you have a question? All really like your presentation, but I really want to, Matthias Kretzler over here. Yeah.
Okay, hi.
Yeah. And really want to hark back on one area where you failed, we failed as a society, we failed. That during the pandemic, we were not able to effectively counteract the misinformation. And yes, you are communicating to scientists, but as you said, a reasonable understanding of scientific history, this was all reasonably predictable. And educating scientists early on aggressively how to counteract misinformation is a charge everyone in this room has as we are educating scientists. And I'm also a physician, so I also failed my patients who died of COVID in my inpatient services and my outpatient services. And really learning how to counteract that and moving forward. We are living and we are here to serve the society. And we have to get our messages out. And you, as a not-for-profit entity, have the credibility in that space more than anybody else and really would enforce and recommend that you work actively with the community, you bring those papers forward into social scientists, how to develop and support technologies, how to do that most effectively.
Yeah, so we've had a lot of pieces on this. I think that, but I also think that reaching the influencers who, the church leaders, the family members, because if somebody, I always say to the people I grew up with who believe some of this stuff. I mean, I'm from Fayetteville, North Carolina, as Gil said. It's the biggest Army town in America, so I'm not in the political majority there. I'm like, okay, do you believe me? Have you known for 50 years? Or some jackass has got a podcast, all right? And what? They believe a jackass who's got a podcast. And so the only person who's going to compete with that is their priest or their local politician or, someone they know that they trust. And, those people aren't reading science. So, that's a tricky challenge that I'm not sure. I want to work on it. I've written many editorials about this. And we published lots of perspectives and review articles about all of this stuff. But, who covers our stuff? The mainstream outlets that these folks aren't watching. They're not reading the New York Times and listening to NPR. And watching the NewsHour. Those are our big outlets.
Yeah. No, but you have a critical multiplicating factor. And yes, you can reach people who can reach people. And this is, our society, American Society of Nephrology, does that exactly. We are charging our dialysis nurses to address these with people in the dialysis units who are the mothers and daughters of the priests in the churches. And this is a societal effort, and you have the ability to serve and continue to serve as a multiplier there.
Yeah.
And that's the only statement. And
sure.
I know you have been on that boat, and I want to reinforce that journey to continue.
Okay. I couldn't agree more. And, all I can say is we're giving it all we got.
So I've one question or it's a question comment about this. I think we need to understand why 100% of Brasilia, the city of Brasilia is vaccinated when they are in the midst of misinformation from their leadership as well. So, so what is it about, these different populations? I think June actually hit on it. It's a cultural thing that the culture can overcome the leadership and the disinformation. So I think it's a very interesting confounding thing. One more question, and then we're going to let.
Hi, my name's Bailey Garb. I'm a PhD student here in the department. My question really is directed at Dr. Thorpe. I wonder, so Science, Cell, Nature, these are without a doubt the most high impact journals we have in the field in general. And I wonder how that creates sort of a bottleneck and almost limits open access. So, in order to get into these journals, you need to have extremely high impact paper, which often takes a really long time. This could be a culmination of 3 papers in a less high impact journal. And that increases the amount of time that labs are holding on to data, holding on to publishable works in order to reach these high impact journals. So how do you combat this in your attempt to be as open access as possible?
Well, you're absolutely right about that. And that's a major shift that will need to happen. It's not, so first of all, I always tell audiences with students, and you reminded me that I forgot today, that if you send a bunch of papers to Science in your career and none of them get published, you can still become the editor-in-chief. Okay. Because I'm O for career sending research papers to Science myself. And, you know, I grew up, as I said, publishing in the Journals of the American Chemical Society, and I was happy when I got a paper into JAX, and I'm proud of my JAX papers, but I'm also proud of my papers that were in inorganic chemistry and JPhysChem, which is where I sent them if they didn't get into JAX. And I made my career doing that. I got tenure and I got promoted and I got grants and I was able to do everything I wanted to do. So, you know, the short answer, which is kind of glib, is, you know, people should send us stuff when they think that it's just so high impact that it's worth all the garbage that you said they have to do. You're right about all that stuff. But in the, you know, in terms of trying to change that, you know, one thing is to really celebrate papers that don't get into our journal. And, you know, we have 6 journals, and one of them Science Signaling, which has an impact factor of 6. Okay, well, we're proud of our Science Signaling papers. You know, there's some great stuff in there. They're long mechanistic papers that explain how things really work. And, you know, I'm constantly pumping up Science Signaling as much as I can. And if you get a paper in Science Signaling, you should be proud. That's a kick-ass journal. And so are, you know, the journals that I just named, or the specialized journals in your field. There's nothing wrong with those papers, and I'm proud of all my papers in the specialized journals, and I hope you will be too. But, you know, what I tell folks that work for me we're trying to do is we're trying to find, unfortunately, we can't pay the bills if we don't find the papers that everybody wants. So of course, when Google won CASP, we called up Demis and tried to get the paper, because that's going to get cited thousands of times and lots of people are going to click on it, and it's great. And we have to have that to pay the bills. But we also want the papers that nobody knows about. So, you know, it's fun for me to come to the University of Michigan and give a talk because there's so much great science here, and this is an amazing place. But I've been to a lot of universities that science editors haven't gone to say, don't think that, you know, it's impossible, that you've got something that we want. And I'm constantly sending my folks out to do the same thing. And we got to try to break down a lot of this ossified gatekeeping that you're describing. But it's not going to happen overnight, because unfortunately, you know, one of the reasons why we have to have journals is because if we didn't, the deans and provosts of these universities wouldn't know who to promote. They can't read all of this stuff themselves and decide if it's excellent science or not. You know, when I was a provost, I used to have this many tenure cases sitting on my desk in the spring. And, you know, I just went and looked at the CVs, what else I was going to do. And the giving grants and the whole, and you all leaving and getting jobs. I mean, I hope your advisors are not going to let you go out on the job market with a list of preprints. I mean, if you're in computer science, you can do that because the way they do their meetings, and that's actually a very progressive system. But for those of you who are in the life sciences, you know, you can't do that. My son's a graduate student. I'm not going to let him graduate with a list of preprints on his CV. And thankfully his advisor's not either. He's not going to do that. So, you know, this whole system needs reform, and, you know, we got to give it all we got to try to change it. And, you know, we have confidence in all of you to do that when even now, but especially in the future.
Well, that's awesome. Thank you, Holden, very much for this wonderful keynote. Thanks to the panel for a great discussion and everyone. Now, there's just a few little things. First of all, you know, Gil, how wonderful it is on this 8th Omen lecture that we got the policy discussion going, which is something, Martha, it's something that we really wanted, and it worked out great. This is a time to expand our minds beyond just the narrow focus of the science of bioinformatics and computational medicine and bioinformatics, and think about the big things. And Holden, you've helped us do that in the 8th Omen lecture, and we're really appreciative of that. Thank you. Well, and you know, the whole day has been so great, and we got more out there in the all the posters and the refreshments and everything awaits us. Before we do that, this has been a great day for our department and for the medical school and the university, and it's something that depended upon a lot of people putting in a lot of hard work. Teresa Nestor, Elizabeth Paymal, you've seen Aaron and Paul and Ken around here. The entire team, our great staff team, did so much. And of course, at the last minute, Ala generously pulled together that wonderful session that was this morning with the students. Margaret was there, and Katie and Julia helped. I mean, it's the big team. It's you didn't see my talk, but I ended with that same statement that you said, Dan. And so, and we talked about the future. And the future lies in the students back there and beyond into the screen and all their faculty and those posters. It's special because we're going to a special place. It's called the Omen Atrium. And we're going to go now visit the Omen Atrium and enjoy our reception and the posters and having those conversations that we can't have on Zoom, but we can have right here now. Thank you so much for everybody's attention. And thank you so much, Holden and all the speakers and guests. And Aaron, what a job. Thanks, everybody. And let's go have some nice reception.
Before you have a hot light moment.